Okay, allow me now to welcome Jean-Pierre Chalain, Deputy Director of Swiss Gemmological Institute. He will be talking about new frontiers in pearl analysis, age dating, DNA fingerprinting, and novel radiographic methods.
Ladies and gentlemen, first of all, I would like to apologize Michael Gemmological, who unfortunately could not make it this time in Bahrain, and I tell you he's very sad not to be present here. In contrast, I'm very happy to be with you. It is my pleasure to give his talk. I hope that you are not afraid with contrast, because I will not be able to make a show like Rui just made. My presentation will be on a more scientific base, and I apologize in advance if you are not that much convinced of the fancy work that we do at the SSEF. But we have a lot of enthusiasm, Nick, as you said in your presentation, and this ran the lab as many labs. So, as you know, the main duty of Gem Labs is to separate natural from cultured pearls. This is an issue for the trade. Just have a look to the difference of price between two similar looking pearls. Although in some cases the difference between natural and cultured pearl is straightforward and easy to separate, sometimes it may be very difficult and challenging. Some other colleagues have shown some different difficult cases, and we have such cases. This is why we are always trying to find out some new technologies that will help us to give you more reliable results. So therefore, the conclusion that we give on lab certificates is always an expert opinion, and our findings are always based on educated interpretation. As most of you know, I am also the head of the diamond department, and when Michael 2 days ago asked me to give the presentation that I just discovered 2 days ago, I thought that where is my legitimacy to give this talk to you now as a person specialized in diamond? And I found my legitimacy on this slide here, because I realized that this is my hands. So I feel responsible now with you to speak about the pearls. Pearl testing in a Gem Lab is based on a combination of many different investigations. Of course, the first thing that you do is looking at the pearl with your sight. Then you can use a loupe and a microscope. You can visualize the internal structures by X-ray radiography or X-ray microtomography, digitalized. You can have access to chemical analysis by such as energy dispersive X-ray fluorescence. You can use structural analysis such as Raman spectroscopy or photoluminescence, to name a few. These are the traditional methods that are used today in Gem Labs. Some novel methods are evaluated at the SSF on a continuous basis to get new insight into pearl formation and their internal structures. And finally, with the aim to support pearl testing as a client service to the trade and consumers. So one of these first methods that I would like to describe now is the radiocarbon dating of pearls. This technology is not new in the field of gemmology. You know about age dating of zircon in sapphires, for instance, but it was a direct radiocarbon age dating on pearls was not available a few years ago. And this method is able to provide supporting evidence for historical pearls and may also assist, you will see some examples, in the differentiation between natural versus cultured pearls. So the C14 radiocarbon age dating method is, I will shortly summarize. Roughly speaking, cosmic rays will transform the nitrogen present in the atmosphere into C14, which is radioactive and therefore unstable isotope of carbon. And as unstable isotope, it has a short half-life time. Its half-time life is 5,700 years only, which enables an age dating up to something like 50,000 years. How does the age dating on C14 works? The C14 is incorporated into living plants and animals, and by breathing, for instance, or eating. And because the C14 decays, if you make the ratio of C14 to the total amount of carbon atoms, then you will have an idea of the date when the animal or the plant died, because it ceased to incorporate the C14. So this is the basis of the method. Applying this technology to Pearl was a challenge because, as the common challenge of gemmologists is to make conclusions with non-destructive analysis, we had to find a way to destroy as weak as possible the samples, and we were able to sample only a few milligrams, something like 2 to 4 milligrams of calcium carbonate out of pearls, and to show that this is sufficient to come to an age dating. This method has been applied now on pearls that you have already heard of. These pearls were found in a shipwreck in the Sea of Java in 2003, and we have been lucky to analyze these pearls by this radiocarbon C14 analysis, and it shows that in comparison with the age of that was dated by archaeology studies on the boat, which was proven to be about 1,000 years old, the C14 radiocarbon age dating came up to the same result, because you see here the age dating on this scale here. So this is a confirmation that the techniques work, even with a very, very small sampling of the pearls. We could also analyze the Anna Maria pearl. This pearl is documented since the mid of the 19th century. It originally belonging to Anna Maria de Sevilla, and it was probably fished during the Hernan Cortes conquest in the 16th century. And the radiocarbon analysis that were carried out by the ETH in Zurich have been date the pearl at the same period between the 16th and the 17th century, and these perfectly match the documentations of this historic pearl. These methods are very sensitive, and it was we applied the same technique of radiocarbon analysis on very recent pearls, cultured pearls provided by Paspalet, So they were produced in the 50s, late 50s, and it was interesting to make the match of this method to its young age, and to compare it with the bump peak, which is a curve that is very famous for people working on age dating by this method, because this bump peak that you see here actually takes care of the increasing C14 concentration in the atmosphere due to the period where there has been nuclear testing in the period of between 1960 to 1970. So this is called a correction curve that we have to incorporate into the analysis to conclude the result. Another correction curve that is not shown here relates to the usage of fuel or oil. Since the industrialization, a lot of fuel has been emitted in the atmosphere, and this fuel is extremely poor of C14 because it is coming from fossilized vegetal organism, and therefore the C14 is not present anymore because of the half-time. So these are curves that are very important to incorporate in the correction when it deals with age dating. I'm sorry? Yeah. No. Please wait. Thank you. As I told you before, age dating is also a technique that may be used to separate between natural pearls from cultured pearls. Here you have a micro radiography made by tomography that shows ambiguous result to interpret. You see the famous moustache that most of gemmologists dealing with pearl do not like much. And it was not clear whether this pearl based on this radiography was a natural or a cultured pearl. And therefore we decided to go to ETH again and make some, based on the same sampling procedure, make some analysis. And the age dating proved that the pearl was from the 17th to 18th century, which proved that the pearl was natural pearl. This service is now available at the SSEF since February 2017, and this is done together with the Federal Institute of Technology in Zurich, in Switzerland. All the papers that you have seen that are quoted in this presentation are all available on the website of the SSEF. You just download. Now, the second technology that is quite interesting and promising on our trade is the DNA fingerprint. The species identification based on DNA fingerprint is a new method applied when it applies to pearls. We will see an example of application on coral as well, and this is a new technology that is coming inside the world of gemmology, and this is a very promising technology. Especially when it deals with CITES or things like this, for a fair trade. This is very important. I have to say that gemmologists are not specialists in genetics. It's not yet in the program of their formation or their education, but maybe in 20 years' time it will be mandatory. Apart from species identification, this method may also assist in natural versus cultured pearl separation, but may also provide information about the geographical origin of pearls. So the principle of DNA fingerprint. Most of you have seen some series where a criminal will be identified by its DNA, because DNA does not only identify a species, but if you go down to the good sequencing, you can also identify not even further to the gender of the person. You can also go to its own identity. And of course, so the DNA is a series of 4 nucleobases, the A, C, G, and T, and they order in different orders, and they form series that we will sequence, and this gives rise to the genome of the different species and different individuals. So the question is there any DNA in pearls? A pearl is mostly made of calcium carbonate, as you know, something like 92%. So this is a crystal, or this is a mineral, and DNA is something that is not mineral at all. So the organic substance that may contain DNA is the organic matter, and there are also some water and minute amounts of residual substances in pearls. Yeah, and what SSEF has shown in 2013 already, and this is published in an open source journal, so it's open to anybody, any organization, any lab, any people. It was this paper is very important because it is the methodology that is used for extracting the DNA out of a pearl. So, and with a very small amount of sampling, such as 2 milligrams only, we could prove, and this is the matter of this publication, we could prove that this is a sufficient extraction of DNA to achieve a positive species identification. The method is based on the fact that DNA is a negatively charged molecule that has some affinity with the C calcium 2+ of calcium carbonate, and this is one good point for the possibility of this technique. Whenever you have extracted only 2 milligrams of material, then you would like to extract the DNA. Extract it, separate it from the calcium carbonate. To do so, you will use a liquid that is called EDTA. I don't give you the full name of it because it's long like this and it's not my cup of tea. But if some people is interested, I have the paper in my pocket. You will let the carbon the sampling overnight in this liquid, and this will separate the DNA from the matrix. Then you will concentrate the DNA with a vortex by centrifugation, and later this is not sufficient. Geneticians are used to first of all identify which sequence they want to study to make separation between the different species. So this is made by using some markers. You will then have to amplify the DNA concentration because out of 2 milligrams you don't have enough material to have a good result. And to achieve to amplify the DNA quantity, they use on some sequences they use PCR. It's a polymerized chain reaction. Sorry, see, I'm not familiar with these techniques. And and this will replicate the DNA in thousands to millions of copies, and then you got sufficient material to come up to a result. So this method has been used by SSEF to separate different Pearl species. As you see some results here with Pintada maxima, radiata, and Margarete ferra. And this is a technique that is new, that is extremely sensitive, and we are certainly not able at the SSF to provide this result on ourselves. We are very fortunate to collaborate with the forensic medicine laboratory of Zurich. These people are this laboratory is ISO certified. They do criminology. They do very high technology. They use very high technologies, and they work on a very reliable scientific basis. And this is why we are very happy to collaborate with these people. Recently, we could also apply these techniques on coral, and again, this is very promising for the trade. Recently, using the same techniques and the same laboratory, I am authorized to tell you that recently we have identified some samples from the trade that are from a species that is not described yet, not even in CITES. So this is the people who are working in Zurich. They are very keen on this new technology applied to gemmology, and they work very, very hard on this, very hard. You have probably seen this paper that Dr. Laurent Cartier, Michael Gemmological, Bertalan Lande, and Johanna Meyer have published in Journal of Gemmology in 2018. This is the same method of DNA fingerprint that applies on not only a coral but also on ivory. You know that the trading of elephant ivory is forbidden today when the mammoth ivory is still authorized. So it is important to be able to make the difference between the two. And gemmological investigation, traditional gemmological investigations, is not always easy to use to make the separation between the two. So therefore, a positive way to separate the two species is the DNA fingerprint. And again, this was published in, this is a service that is offered to our clients since 2 months ago only. And it is, the publication again is on the SSF website. And you are welcome to download any of these papers. The last method that I would like to propose you is, let's say, ongoing technology. We are investigating in these new techniques to analyze or to better understand what are those contributions to the gemmological field. There are two different techniques, the X-ray fast contrast and the X-ray dark field scattering, and also the second one is the neutron radiography and tomography. Both approaches visualize internal structures in pearls and may be used complementary to the classic X-ray radiography that you have shown, that are shown here. So this is a traditional X-ray radiography. On the left-hand side, you have a beaded cultured pearl. On the second on the left is the beaded cultured pearl with a natural pearl as a bead. You can probably see the radial structures of the calcite at the center of the bead. The third from the left is a beadless cultured pearl. and the last one is finally a natural pearl. So these are traditional X-ray radiographies. You see what we like as gemmologists when it deals with identification of natural pearl. We like to see these radial calcite columns. This is shown on the left-hand side photo on a section of a pearl, poor pearl. Yeah, but we are lucky to see what it is actually. Because we are not allowed to sew all the pearls of our client to find out the structures, then we have access to radiography and also micro radiography. Can you please run the video? And this is the scanner that we obtained by. So we are gently entering inside a pearl from the top to the bottom, and we will reach very soon the center of it, where you will recognize this radial structure from the natural pearl, from the calcite crystals that are typical. Here we are. Okay, yeah, we're very, very happy to see this. And we are going out of the pearl from the bottom of the pearl. So this is a scanner. Huh? Here we are. Okay. Thank you very much. Now, complementary to this technique that is available in most of gem labs, we are now investigating in new technologies such as X-ray phase contrast and dark field images. This technology is based on two moving gratings that create an X-ray interferometer, and this gives rise to not only the normal absorption radiography that is shown here, but also to two additional techniques or means that give quite different imaging. And this is what we are investigating at the moment. So the second one is the refraction. So you see that the images are quite different and may provide some results that are not visible by the first method. And the scattering image is also something very promising, where you can see some features that are absolutely not visible in the other techniques. These dark field images and contrast imaging have been applied for you on these beadless cultured pearls. You have the different results here, and you probably understand that this result is quite interesting when it deals with, if this is the same pearl, and this last technique is quite interesting for us. You have also the image of the dark field image of this beadless cultured pearl freshwater from China, and it is a very nice imaging, but we think that this is a very promising technique to investigate. And also these techniques enable us to go to the scanner, to the X-ray radiography, microtomography. And this is quite interesting as well, because it will provide us with some new insight in and viewing to better analyze these pearls. The very last techniques that I want to show you is based on neutron imaging. These techniques has been investigated by SSEF in collaboration with the Laboratory for Neutron Scattering and Imaging, the Paul Scherrer Institute, PSI. They are using a synchrotron and neutron spallation source. They are based in Villingen in Switzerland, very close by to Basel. And I will not go deep into the detail of the techniques because it is not. I'm far away from being a specialist for these techniques, but roughly speaking, they will use a neutron beam that they will slow down this beam by collision with nuclei of heavy water, such as deuterium, to monitor the neutron beam for imaging analysis. The advantage of this technique is probably very well shown on this photograph of a camera provided by Pearl Symposium Institute, where on the top you have the neutron imaging of a camera, where you can see clearly the organic rich material that is absolutely not visible on a traditional X-ray radiography. You can even see the film that is inside this camera, which is absolutely not visible by a traditional X-ray radiography, and this is because this neutron imaging is very sensitive to the presence of hydrogen or boron, and therefore in organic materials. And here is this neutron imaging applied onto a cultured pearl, a beaded cultured pearl. So you have here the radiography, the traditional radiography of the pearl, and the neutron radiography, and you see that the techniques of this neutron imaging is very sensitive to organic rich material, and we make some features to appear when they are not visible at all with the traditional techniques. So this is something that we are investigating, but it seems that these techniques are very, very promising. You can apply the same neutron imaging on natural pearl, on this pink data radiata. We are in the country of pink data radiata. So you have the comparison of this same pearl by X-ray radiography or X-ray microtomography, and now you have the same pearl seen with a neutron radiography, where you see that the hydrogen-rich organic substance is clearly much more visible now on this neutron imaging. Finally, we are happy to show you this fascinating natural pearl provided by Paspere, pink data maxima, of course, and it is fascinating to see this pearl with the neutron imaging. This is for fancy. Again, this has been published recently, and this paper is also available on our website. As a conclusion, I would say that these three techniques that I have been happy to describe, radiocarbon age dating, using a very, very small amount of material, so it's a quasi non-destructive method. The DNA species identification using as well a very small amount of material, quasi non-destructive, is also a new method. The new imaging method that I have shown, such as X-ray phase contrast dark field radiography and neutron radiography, all of these three techniques are certainly techniques that are promising, very much promising for the future of our trade, and we are very convinced that they will support the trade for the future. Nevertheless, never forget that pearl testing is still a matter of expert opinion, and And finally, I would say that scientific researches on Pearl, their formation, their internal structure, and fascinating topics which require multidisciplinary approach involving mineralogy, biomineralization, biology, forensics, and the latest imaging technologies. I would conclude to say that yes, we are working on a fascinating trade. It is our passion, but still we need to investigate, to correlate our results. I'm thinking about the DNA methodology. When it deals with DNA methodology, it is, we have learned with the people from the forensic medicine laboratory that any investigation, any DNA testing needs a positive test and a negative test to make sure that you are looking at the proper sequence of the DNA, to make sure that the proper sequence of the DNA that you want to analyze is correlating both with the available database of DNA and also with your own oysters references. So SSEF has received from all over the world all the species, the oysters with the organic material of the oyster together from all the species from all over the world. And this is our database, and we combine this result with the database that is available online in the genetics field. So thank you for your attention, and I wish you a very pleasant end of the day.